Aloha, everybody. Thanks for joining us tonight. We're going to give a couple more seconds for people to pile in from the waiting room, and then we'll go ahead and get started with tonight's talk. All right. Well, mahalo once again, everyone, for joining us tonight. My name is Sarah, and I'll be the MC for this evening. And this month, we are joined by Dr. John Burns of the University of Hawaii at Hilo and of it's the Multiscale Environmental Graphical Analysis, or MEGA Lab, so MEGA Lab. And tonight, Dr. Burns will be sharing just how the MEGA Lab uses cutting edge 3D technology to map and study reefs through their Map to Adapt initiative. So I didn't know this prior to talking with Dr. Burns, but less than 20% of our oceans has ever been seen. So to better prepare us for the future, we need to know what we're working with so that we can figure out where we need to go. And this Map to Adapt initiative aims to map 1 million of the world's best reefs by 2030. And along the way, Megalab is also working to help equip 10,000 citizen scientists so that they are better equipped to support their communities. So if this is your first time tuning in, welcome. We are the Maui Nui Marine Resource Council, and we have been working for healthy reefs and abundant native fish since 2007. Uh, Obviously, living on an island, everything is connected from Mocha to Makai. And so we work on a variety of projects because, as we know, what happens on land will ultimately make its way to our oceans and out to our precious reefs. So our work takes a variety of forms, and that means doing many things like testing water quality at 41 locations across Maui. Our dive team regularly performs fish and reef survey dives. We monitor the populations and the health of the reefs. And recently, we've also started to tackle some land based issues. So we have provided training on natural alternatives to toxic herbicide and pesticides, most commonly found in landscaping procedures and practices. So um, that's just a real quick snippet of all the things we do, but tonight's presentation is done in partnership with so many organizations, sponsors, and partners, and we want to mahalo the County of Maui for being our partner for tonight's presentation. Being able to bring this all to you is through a partnership with them. And as a quick side note, if you haven't already, you can sign up for our monthly newsletter, The Reef and Brief. It'll bring you more information on talks like this, events, and just projects that and work that we're doing around the island from Maka to Makai. So mahalo to the County of Maui and to all of our amazing businesses and foundations, just so many local partners working together for Maui's marine environment. Mahalo to all of them for being a part of the solution. Um, and so just a few notes before we introduce Dr. Burns tonight. We will, as usual, leave time at the end for Q&A. So throughout the presentation, please use the Q&A button at the bottom of your screen to submit any questions. Unfortunately, due to some technical craziness, we weren't able to stream to Facebook tonight. So we usually have a streaming there, but we'll go ahead and we'll post the recording after tonight is all done. So please hold all of your questions to that box, and we'll go ahead and make a list and talk to Dr. Burns once he's done. And so now for our speaker, Dr. John Burns is a research scientist who studies coral health and disease. His work has investigated the physiological impacts of disease on affected corals and how coral mortality affects the ecology and biology of coral reef ecosystems. John has been continuously searching for both global and localized drivers of coral diseases in order to promote healthy ecosystems in the face of global stressors such as climate change. His expertise led to his inclusion in the award-winning documentaries Chasing Coral, Reefs at Risk, and Island Earth. John has recently developed innovative techniques to create three-dimensional maps of coral reefs to accurately measure how natural and human-induced disturbances impact ecosystem function. John's research has been presented at scientific conferences around the globe, and he has authored multiple publications in international peer-reviewed journals. Megalab also, I would just like to say, has an amazing website and social channels, so check those out if you get the chance. But Dr. John Burns, thank you so much for being with us tonight. The floor is yours to take it away.
Thank you. Appreciate it. Thanks everyone for coming for this talk. Just gonna pull up my slides. Sarah, I just want to confirm, is that working with the
Yes,
full?
that's
Awesome.
perfect.
All right. All right. Yeah. So again, just thanks everyone. I appreciate you being here for the talk. I'm going to speak tonight about unlocking the mysteries of biodiversity on Hawaiian coral reefs. So I'll be giving some overviews of the type of research that we're doing, as well as kind of putting it in just general context of what it means and what we think we can do to try promote having healthy, functional reefs in Hawaii and throughout the globe going into the future. So just to start, I want to briefly introduce the Mega Lab. It's a collaborative group of scientists that began with myself and doctors Haunani Kane and Cliff Capono. And we wanted to basically try to do science a little bit differently. And so we try to train and work with individuals who see the world creatively so that we can make new way for scientific experiences. And what I mean by that is that often science can be a place for discovery, but can be somewhat rigid in its approach. And it can seem to people like you have to act or be a certain way. And we really want to show that it's the individuality and creativity of people sometimes that can bring a new lens to looking at problems and help us discover even better ways of learning about the world, but also protecting it. And if we do that in a vacuum and don't share the work, it's hard to really feel the impact is realized and reaching as many people as possible. So we share a lot of stories that try to explain what we do in a fun and engaging manner, and also redefine how a scientist looks and acts. And behind all of this, which a lot of what I'll speak tonight is in regards to, is the technology that we develop to inform climate action, preserve our ecosystems, and at the end of the day, we want to promote positive human environmental interactions. We're all from and part of the environment, and I think the best way for the future is we find the most beneficial ways that we can interact with it and be a positive partner to the world, which is where, you know, we get everything we depend on. So this is just an example of what the work looks like that we do. This is one of our PhD students, and this was on a trip to Fiji. And just to put it in context, you know, we feel so fortunate that we go out and we dive on these beautiful reefs, and we use cameras like the one that you see here, and we essentially try document and map it. And why mapping is so important to us, as was mentioned in the introduction, is there's so much of the world's reefs and the oceans we've simply never seen. And so we go to places and we try to look at the intricate formations that these corals make. But we do want to also develop mechanisms for citizens to get engaged in mapping so that really everyone can be a part of this. And if we all work collectively together, the more we map, the more we understand, the more we're able to make positive changes to protect our reefs. So in a basic breakdown, when we go to a reef system like this illustration is showing here, we typically go down on scuba and we take notes, we take images, we collect water samples, tissue samples, whatever it might be, and we try to understand things like, why are these coral healthy? What maybe makes them sick? How do they interact with the corals around them? And at the end of the day, corals are forming the foundation of these ecosystems. They're like the the framework and the infrastructure of a city that is what supports all the hustle and bustle and the activities that resides there. So we're fully aware that without the fundamental basis of corals creating and growing as a living organism to make these reefs, we're going to lose, the function of these ecosystems. What I developed during my PhD was a technique that uses a tool called photogrammetry, and it's quite complex, but basically we swim over the reef in an overlapping fashion with cameras like this. We collect a bunch of images, and from those images, we're able to build three-dimensional reconstructions. And I just want to share this quick video with you because, again, speaking on how we want to share what the science looks like, this is like a behind-the-scenes montage view of one of our larger scientific endeavors. So this is on a NOAA ship. We were working down the entire coast of Hawaii Island on the west side of the island, from the most northern part all the way down to the south. This was a collaboration with students and scientists from UH, NOAA, Division of Aquatic Resources, and our goal was to do the three-dimensional mapping like I talked about, and in the exact same places we are mapping the reefs three-dimensionally, we were also doing visual fish surveys, and these pumps and devices that you see in the lab, that was what we were using to also collect water samples and then collect eDNA. And so ultimately, we are trying to understand, what is it that's driving biodiversity, and how can we use these new tools to understand what's there and what role these coral reefs play. And so previously, you were seeing footage of us behind the scenes on the ship. This is what it looks like when we launch these small boats. And it's an amazing amount of planning that goes into this. We spent over 2 years designing the approach and picking our sites carefully so that we could represent all different types of reefs, from sand to boulder habitats to tons of live coral. And here we have some of our students. It's pretty cool. In the picture I saw early in the intro, I'm pretty sure I saw Lily Gavigan there in the beach in the water quality thing, and she's one of the divers here. And so her and these students, here they are, they're swimming over the reef, they're collecting all these images. And so this is what it just, physically looks like when we're out there. And it's years of planning that went into about 2.5 weeks of work. And each day is extremely intensive. We're up early. We're out diving as long as we can, basically all day, doing 4 or 5 dives a day. And we have several different teams on these small boats. So after the three-dimensional divers go out and collect images to understand the habitat, fish divers go out and visually assess fish. So basically looking, counting, marking everything they see. And here you're seeing some NOAA divers grabbing water samples. And these are what's used to gather eDNA. And so essentially all the fish, they have mucus, they have scales, they're peeing, they're pooping, and all the stuff that comes off, it leaves a DNA signal, just like, you see on shows like CSI. And from that DNA, we're able to figure out exactly what's on these reefs and give us a view into the darkness, essentially, of the fish that we don't see, like nocturnal fish or cryptic fish. And so ultimately, we're piecing together this picture of, what's really going on, what's there, and how does a healthy coral reef play a role in all of that. So hopefully that just was somewhat enjoyable, and it gives you like a little bit of a sense of seeing what it looks like when we're out there doing the science. So going back to the 3D monitoring, this is a really cool technique that came out of advancements in data science. And essentially, in a reef plot like we're doing, we're surveying areas about 20 meters by 5 meters, so about 100 square meter area. And we'll collect several hundred very high resolution overlapping images. And modern cameras have metadata, which essentially tells you the type of lens you have, the focal parameters, and it's not too dissimilar from your brain, in the sense that when I'm looking at any object, my brain tells me how far I am from it, and I'm able to sort of understand the size and scope of what I'm looking at. And you can do the same thing with the images, because when they overlap, you're getting multiple views of a scene. And a good analogy of this is like birds. If you're ever sitting around a park, birds are flying by looking for your crumbs, and their eyes are on opposite sides of their heads, but your eyes are like cameras. And so when the bird twitches its head, which if you watch, you'll notice they twitch all the time, and it's giving them a different view of the scene. And so their eyes, like cameras, get multiple images of it, and then that lets them reconstruct depth. And so we feed these images through pretty sophisticated like algorithms, and we're able to reconstruct them into what looks like a full three-dimensional reconstruction of the reef. And then from that 3D reconstruction, we have in the top right, this is just an illustration representing it, we get like a stitched photo, almost like a panoramic photo of everything that's there. And that allows us to go in and mark what corals are there, what disease they might have, what species they are. And every pixel of it contains XYZ information. So we also know how complex all of that is. And this video is just giving you an example of what that looks like. So here's a reef plot, and you can see all the corals. And this was a massive advancement because prior to this, all we were able to do was just take photographs, right? And a photograph is nice, but it's just one single point, one spot. It's hard to understand from that intricate features of like what type of structure each coral is making. And so from this video, you're able to see all these different species. And if you notice, right, it's such high definition, you can make out little things like algae interacting with the corals, white spots where the coral might be sick or growing. You can see areas where it's dying. And so we're not only able to get a snapshot in time, but we're really able to like virtually reconstruct exactly what that reef is like at that moment and understand how each living coral itself is providing habitat. And this was such a huge motivator for me because when I began studying coral diseases, I remember having folks from agencies ask me sometimes, well, it's bad that these corals get sick, but like, what does it mean? And I realized that I need to find a way to connect the habitat that each of these corals produces to the organisms on that reef and actually understand the linkage between habitat structure and how that influences the fish in that space. So this is another graphical illustration. And hopefully you can now imagine, like from those reef constructions, we have an image, right, of our study plot, and then we can trace every coral. So in this case, all the reds would be one species of coral, all the yellows would be another, and the blues would be something maybe like algae. And from that, we're able to figure out using advanced statistical tools which ones of these are facilitating certain forms of habitat structure. And the types of habitat structure that we're interested in are things like slope, which is relief essentially, and slope gives fish places to hide or to use to hunt. We get vector ruggedness, which is like an irregularity measure. So branching corals will have more ruggedness than, say, a smooth mounding coral. Curvature on the bottom left, that tells us about indentations, like concave and convex space. So if there's a lot of curvature, there's places for like puhi to hide and juvenile fish to take refuge. Surface complexity is like a general measure of how complex three-dimensionally is this reef, right? Like if we compare the contour to the flat distance, the more complex it is, the higher that number will get. We can classify these morphologies too in their common distinctions, such as, is it a branching coral? Is it a mounding coral? Is it an encrusting coral that just grows along whatever it's on, like a rock, for instance? And we can look at their 3D surface area. And so ultimately, as I mentioned, we're trying to understand how does this reefscape, the city of living corals, influence all the fish and the predators that are there? Because collectively, this is what makes a reef function, right? You need a whole food web, and beyond just having a food web, we need energy to transfer from it. So we need fish that eat algae, fish that eat corals, we need fish that eat those fish, we need larger fish that eat those fish, and if we have that, all the energy moves through the system, and it's healthy and it's productive. And when it's productive, we have the services we depend on, right? So I'm sure most of you here know, right? Reefs are some of the most valuable ecosystems on the planet because they produce oxygen, they give us food sources of fish, and then they also protect our coastlines among a myriad of other resources that we extract from them. And so to understand their relationship with fish, what we do is we break them into functional groups. So we can look at like corallivores, herbivores, butterfly fish, and breaking them into groups helps us understand what are these linkages between habitat and fish populations. And to do this, first we look at how does each type of coral influence those habitat complexity metrics I was describing earlier? And what we've been publishing the last few years is really exciting because we're able to see patterns like mounding and knobby corals produce a lot of complexity. Branching corals produce a lot of ruggedness and slope. And we can then take these metrics and analyze them along with fish, and we see that a lot of them are positively influencing both the abundance of fish, but also the biodiversity. And then we can even go one step further. And I know there's a lot of complex data and graphs here, but I'm just going to try explain it in a simple manner. Hopefully it makes sense. But if you have any questions, feel free to ask me after. And so when we break them into these clusters, which are like specific functional groups, we get an even more advanced picture of what's going on. And these have been some of the publications we've done recently that we're very proud of, where we can now figure out exactly what types of corals are key drivers that support specific functional groups. And we need to know this because when we're managing reefs and now when we're restoring reefs, without that guidance of knowing what type of coral and what type of structure we need to support fish, we're sort of in the dark. But if we know what pieces of the puzzle need to be there for it to be a healthy reef, now we stand a better fighting chance of being able to ensure that we have these features that the fish need and therefore depend on, and then everything is able to be productive and we all benefit from those services I was describing earlier. And one thing that surprised us too, here's like a picture of like a very large reefscape. On the right, you have the photo mosaic. So these are all the photos stitched together. On the left, you have the digital elevation model. So these are essentially pixels indicating three-dimensionality. So the more yellow it is, the higher up it is, the more green, the lower it is. And so you can sort of see that structure. And we found, surprisingly, not only does structure influence things like fish biomass, but it also seems to be related to nutrients and biogeochemical cycling, which does make sense because if fish are residing and spending time in specific areas, they're also cycling the nutrients that are coming from the water, from submarine groundwater discharge. And so this is just giving us more and more clues of exactly how this ecosystem is able to work and do what it's supposed to do to be healthy and productive. And here's a few graphs to just show specific things that we were impressed by, where like we can figure out exactly which type of structure, like ruggedness, drives and how it influences corallivores, so specific groups of fish. Piscivores, which feed on fish, they also respond to these, and curvature. Herbivores respond really strongly to ruggedness as well as curvature, and even nutrients seem to respond positively to complexity. So it's not just as simple as, okay, habitat complexity is good. We're able to figure out like what are the players in the game that drive complexity, and then in turn, how is that complexity supporting healthy fish populations? Here's another example. This is sort of a complex ordination from the northwestern Hawaiian Islands. We've been doing this all throughout the Hawaiian archipelago, as well as other locations in the Pacific. And it's a lot to understand this statistically, but if we just break it down, what it tells us is that these colors represent different species of fish that are dominating the reefs. And we can see that certain fish, like parrotfish and herbivores, some of these species prefer deep sites with certain levels of complexity. Some of them, like wrasses and other parrotfish, like really high complexity. Some of the smaller fish and surgeonfish, for example, and wrasses, they tend to be dominated at low complexity sites, as well as shallow sites. In addition to all of that, coming back to the eDNA, this gives us a chance to do something we've never done before, which is say, okay, in addition to what we can count, which is only what we can see, what species are there that we're not able to see? What are we missing? And like I alluded to before, this is really critical because we're so limited by time and resources when we're out on that ship, right? And we can't scuba dive at night, and we only get to hit each spot once, that we miss things like cryptic fish, eels that might be hiding in holes, large predators like sharks and aluas that can come through the space very fast, as well as dolphins, and also nocturnal fish like manpachi and other ones that are out at night. So when we collect those water samples, we're able to filter the water down, amplify markers. And so this is the same as like a DNA paternity test, right? Each marker it can be used for all the different species. And so if that species has been present, we will detect it. We can then sequence it. And from this, we're able to identify all the different species we're seeing. And beyond that, because we have the visual surveys to sort of cross-reference, we can take the number of reads that we get, meaning the amount of DNA that is there. If there's a lot of DNA, we'll get a lot of sequence reads. If there's a little bit, we get a little. And so we can even from the DNA infer how much of that species is there. We can get an estimate of biomass. And all of that just tells us what's going on in a much more clear picture for these reefs. And so just to give some specifics, if you're curious, from one of these reef plots, like you see on the left, we'll get about 24 liters of water. And then we use all these different primers, right, to detect what's there. So we can get metazoans, fish, general eukaryotes, cnidarians like corals, and also algae. And so this is a very, very large, very collaborative, ongoing project, but it's really exciting because to date, it's the largest marine biodiversity assessment in the world using eDNA. The ocean is a wonderful place because it's nutrient limited, so DNA breaks down fast. So it's nice because we don't get DNA, traveling miles and miles and miles. So it's not like you'll pick up a signal of something that wasn't there. It's pretty accurate. And from this, we're already noticing in the preliminary analyses some really interesting trends where there's certain regions of the coastline that seem much more productive and more important for supporting biodiversity. And you can imagine how valuable that information is for state resources and managers, where if we have to decide where to protect reefs, where to make MPAs, et cetera, this is the information that can guide us. This is what can tell us which areas might be at risk, what type of reefs might be very healthy. And without that information, you're just limited in your ability to protect and conserve them. And so in the next, year or two to come, we'll be publishing a lot of this data. But what was really impressive is we found hundreds of species of fish, of algae, 33 species of corals, and over, thousands of species of plankton, fungus, and inverts, microbes. And many of these we just were never detecting in surveys. So our overall understanding has just gone leaps and bounds to what it was before. While all of this is great from a research context, we also want people to, again, feel like they can be a part of these reefs and that they can see them. And so a few years ago, this was really a project for fun. Honestly, I knew that a lot of scientists would give me a hard time because we're all about replication, right? So in that large biodiversity study, we're looking at over 100 sites across the whole coastline, and we want replication, right? So that we have accuracy in our data, that we're confident in what we find. And so with a camera, you've just got one spot. But I had a feeling, that people would like it because you can just watch the reef. And we also get to see how organisms behave when we're not there. And the reception has been kind of amazing from the public. We've got over 11,000 subscribers, millions of views, and people from all over the world have reached out to us from, schools, educational groups, all the way to people we didn't expect, like hospice facilities have been using this because people find it so peaceful. Individuals with disabilities that can't go in the ocean have said, how incredible it is for them now to feel like they can get down on the reef. And what I mean about seeing exciting things that we typically miss, this video was from like the third day the camera was in the water. And right away we caught octopus, mating on the camera. If you look closely, you can clearly see the one in the front of this coral. But if you look behind, there's another octopus there. And we were just blown away, and since then we've caught octopus mating multiple times. We've seen sharks, aluas, monk seals, all kinds of creatures that we've never seen in the 10 plus years we've been surveying this location. And so it's, kind of rewriting what we thought we knew, and also helping us appreciate more that, look, there's limitations to what we can understand when we're diving because we're loud, we have bubbles, we scare things away. And so I think these kinds of technological tools can really help advance our understanding of these reef systems and get a better a better picture of like what these fish are doing really, when no one's watching. we joke that it's like only fins of marine organisms. This was a hawksbill turtle that had never been seen before, so this went into the hawksbill database, and they added that one. And I also wanted to share, this was just today. I thought it was kind of funny that this popped up the same day I was doing this talk, but if you watch on the left side of the screen, you should see a big, large organism here coming your way. And this is a large monk seal, and again, there's not a lot of monk seals out now, and so just to be able to capture them and see where they're going and how shallow and inshore they're coming is a real great resource. But beyond just seeing things and having access to this live stream, which I should mention, it's on YouTube, streaming live every day. It thankfully survived all these crazy west swells we had this winter, but it was hard for me to watch. It was shaking around. It looked like a blizzard. But we're also able to use data science tools to then increase our capacity to monitor reefs. So in this video, we've trained AI to recognize different fish species, and it's unbelievable to us how accurately it's able to do it. And so by combining this continuous camera feed with these AI tools, now instead of spending all this time and money and resources to go scuba diving, which I love, and there's always going to be a purpose for that, we can get so much more data, though, just by leveraging the tools that are at our fingertips today. This graph is just showing, again, it's some complex stats, but what I want you to focus on are these ellipses on the left. And they're showing what we see when we do a conventional scuba dive that like NOAA does, the state does, that we do as researchers, right? We all use standardized methods so we can compare our data. And that is in black. And then if we do very similar surveys with the camera, like we basically made the camera, because it'll spin around and look in all directions, we made it spin around in the same time frame that a diver would when they do a survey. And we had individuals just watch the video and count and size fish the same way they would as if they were diving. We also had people snorkel in green and free dive in purple. And what we didn't expect is how much they all overlap, which is really great news because it tells us, A, there is a utility for these cameras to be serving a purpose in surveying and understanding reef fish. Two, free divers can actually contribute really strongly to surveys. It doesn't just have to be scientists on scuba. We can also solicit help from the public, right? From people that free dive and people that snorkel. And on the right, if you're curious, this is just showing from any given survey, like all the different types of species that they're detecting. So we're able to very specifically say which surveys are best at picking up what different types of fish. So while it's very methods-based, it's a way for us to sort of audit our approaches and figure out, what's the best way we can do this to understand these reef dynamics. The last science thing I want to talk about is just some of the molecular profiling we do. So in this picture, there's a large brown rice coral in the middle. If you look on the left, here's one of those mounding corals. This is a Parietes coral. And in the middle, there's these white lumps. These are diseases that grow on rice coral. And there's also another purple rice coral. So we've got multiple species of corals all interacting. And we like to try and understand, what does that mean on a chemical level, right? When they're interacting and fighting for territory, is that good? Is that bad? Are they, depending on certain chemicals and signals, what can we learn that could tell us about biomarkers of reef health? Because if we can figure out chemicals that indicate a growing healthy coral versus a diseased one, we now have tools to sort of assess and answer, hey, is this reef or is this coral healthy? And this is the same thing that people do when they go to their doctor, right? You say, hey, how am I doing? And they take blood tests, they look at markers in your body, and they say, okay, well, you have this much of this, and that kind of indicates what your general health status is. Otherwise, it's just all relative, and we don't really know what's going on. And so we sampled across these boundaries where corals are competing for space, and we sampled disease coral, and then we can run them through mass spectrometers and essentially analyze all the chemicals. This is difficult, right? I mean, what you see is confusing to anyone that looks at it. It's just peaks that represent molecule size. So we take those patterns, we put it into networks, and then from these networks, we see similarities, and we can figure out exactly what chemicals are on these reefs, like tryptophan, thymidine, lyso-PAF, and then we figure out what they are, right? And so some of these are essential amino acids, which are important for energy, bioactive lipids, which help corals have mucus that keeps them healthy, as well as positive bacteria that is just like our, gut microbiomes. What helps us digest food, helps us stay healthy. And what's nice about combining these technologies is we can overlay it on those 3D reconstructions like I showed you before. So not only did we sample these corals, but we also three-dimensionally map them. And that gives us, again, this visual baseline of saying, okay, where did these pieces of data come from, and how can we overlay them back onto this coral assemblage to better understand, what dynamics are in play here. And when we visualize that, we get to take all that complex molecular data, everything from the spectrometer to the data science pipeline, and boil it down to such a simple thing of a color pattern. And so what I'm doing here is just cycling through different molecules. And if they're in red, they're in high abundance. If they're in yellow, they're in low abundance. And so now we can take this wildly complex data set and just look for differential things. Because in the middle is diseased tissue. And so whenever we see something like thymidine that's in higher levels in disease, but lower levels everywhere else, we now know, okay, we have a potential biomarker here. We have something that we can use to go out and say, hey, is this thing healthy or is it not? And again, this is all relevant to management. When we're trying to assess reefs, we have to develop better mechanisms than just simply looking at it with the naked eye. Otherwise, all we're doing is reacting to problems. But if we can get ahead of these problems, we can hopefully find out conservation tools that help us keep reefs healthy. So to do all of this, we need engagement, we need people to care, and we need people to understand how science can improve their daily lives. And so we are very passionate about outreach and media and education. And so in downtown Hilo, upstairs in the Mokupapapa Discovery Center, which is a federal science building that showcases all the reefs and resources in the monument, we were able to build a venue space, and it's open to the public, and we have an augmented reality app. So students, kids, the general public, they can come in every single day and they can interact with these data products in their own way. And everything down to like the Mega Lab sign that you see up on the stage, the photographs that we have hanging, all of them are targets, like from we borrowed it from that game like Pokemon Go. So kids can come in, they can pick up a tablet, and anything they pointed at will either pop up like 3D models they can interact with, or video clips from some of the documentaries we've made, or websites with information, so they can sort of choose their own adventure and learn about reefs. And then we try to host quarterly events where we just have, we have fun science is the way we like to put it. So rather than kind of, and I apologize if I did this evening, but rather than try throw too much hard or potentially confusing data, we just try to get people talking, having conversations, and again, understanding why do we need this science? What is it doing for our resources? How can we help keep our islands and also the global ocean to be a healthy and better place? We showcase some of the movies we've made, like Mapping the Reef Cloudbreak. We've been partnered with Reef Footwear, and we've been traveling around the world to surf breaks to actually map how reef structure influences waves and what kind of issues are going on there. And last year, in addition to this, we went to Tahiti before the Olympics because the communities there were very worried about the development they were doing for the judging tower. And so it just gives us a chance to respond to concerns that are happening on the ground, to issues that people are dealing with their reefs. And, for an example, in Tahiti, we trained everyone there, this whole community at the end of the road at Teopo'o. We trained them to 3D map. We gave them all these GoPros and cameras so they could do it. And we were able to publish a paper showing that, look, that reef, all those live corals, is value over $1 million. And so when those damages happen, there's a massive economic impact. And even more importantly, beyond that, this is the resource that these people's livelihoods depend on. So we're really trying to continue this work where we don't just do science to do science, but try do science with a purpose that can help these ecosystems and also help people that depend on them. This is just some of the summer events that we do. If you're interested, we've been running a camp in partnership with East Hawaii Cultural Center and the Pangea Seed Foundation called Youth Artivists Hawaii. We bring in a bunch of like middle school age kids, and Pangea Seed brings in these renowned global artists. Up there on the top left is Sinza. He is from New Zealand. And so the kids learned all about marine science and reefs, and they, talked about what they thought to be important, and they used that to produce a mural. They also had their own band. They threw a killer like rock party for all their families and friends at the end. And we had this beautiful mural on display for the past year. And if you go now, you'll actually see a mural done with the kids in Punky Aloha, and that one's going to be up until next year. And all of this really is, like I said, to preserve coral health, right? We need healthy corals because these healthy corals are what all the life on these reefs are depending on. And if we have healthy corals, we have healthy islands. When we have healthy islands, we have healthy people. And we came from these environments. I really believe that. And if we can't sustain them and be a functional part of them with positive human environmental interactions, we're going to lose them. And it's just a sad thing to think about, because if we lose those resources, it impacts our livelihood. So we're hoping to do everything we can to not just keep innovating and advancing science, but find ways to make everyone a citizen science, get everyone involved, and hopefully have them see the value of, let's all go in this together and let's do everything we can to have the best reefs possible, because it benefits all of us. So hopefully something in this talk was interesting for you. If you have any questions, I can take those now. You're also welcome to email me. You can check out our website, our social media, and the YouTube channel if you want to watch the live stream. So thank you very much for having me. I really appreciate your interest in our work. And yeah, I hope you found something interesting from this talk.
Thank you so much, John. That was a bit. You did a great job of putting so many amazing things together in a short period of time. And we do have a few questions if it's okay. And I did answer one during your talk. So one of the viewers asked, what's the name of the live, I believe it's you meant the live stream, the live underwater site. So just so everybody knows, the link to that live stream is in the chat. So you can go ahead and grab that link from there. It is YouTube. It's on YouTube. So if you guys just search, like, I think it's just Mega Lab, it should come up. Okay, and let's see. So we have, okay. So to what extent is the eDNA work being utilized in the state's Holomua initiative, which was, I guess, formerly the 30 by 30, in reference to your comment about establishing MPAs, etc.?
So at this point, it's still in like the data analysis and processing phase. But our goal is, to make all of this data publicly available. So ideally, right, we'll be able to figure out these connections between certain types of habitats. So when we went and we had to pick our locations, we needed to stratify the reef, meaning like we needed to know how to classify it. So it's sometimes as simple as, sand is one type of habitat you'll find in the near shore, right? Just a sandy bottom. Crazy live coral cover, kind of like the image, Sarah, you have behind you, right? Like that's another type. And then you've got all these different kind of, realms in the gradient between that. And so we were able to define them on things like their depth, their latitude, exposure to development, things like this, right? And so because we have that information, we're going to know now how those characteristics are influencing specific species. So once this is all done, all of this gets turned over to initiatives like the Holomua initiative, right? And so then they can say like, okay, look, scientists know these types of corals or these types of habitats, they're really critical for, say, minpachi, right? A really important food fish. Or maybe opelu, right? A really critical bait fish that is also used in commercial fishing and really important for coastal resources. And so what we hope is that it just can be a guiding factor where if people know specific fish they want to protect, like parrotfish, all these things that are both consumed but also play critical roles it can just help guide that. And I don't know, I hope that makes sense, but it basically just gives roadmaps where if you want to protect something, now you have like a blueprint of saying, okay, we need to make sure it's this type of habitat we set aside and protect if we want to keep those fish safe.
Awesome. Thank you. Okay. Several, same question in different formats. Are you guys going to ever come to Maui? Or
Yeah,
is there, where is your work primarily focused geographically speaking, and is there potential to expand to other areas or islands?
yeah, just because I'm in Hilo and on the Big Island. A lot of our work is on the east side here, and a lot of it in Kona, down the whole coast. And then we have a lot of projects that are very collaborative with agencies. So the last two maybe 3 years, like we've been training DAR, National Park Service, other agencies to integrate this 3D mapping into their monitoring. So it's really cool for me because when I did this as a PhD student, everyone thought I was crazy and they're like, it'll never work. It's way too hard, way too computationally expensive. Why would we do it? We do what we do now. Why would we change things?
Gosh.
And it's really awesome to see like NOAA, all of these agencies, now it's the standard approach. And so in a way that gets it everywhere, right? So all the surveys being done by NOAA throughout the islands, all the ones being done by DAR, they're all using this approach. And everything we develop, we hand over to them. We give them all of our scripts, our stat stuff, everything we can. We want all of our data to be open source so that collectively across the state, the capacity goes up. And we've had a really great relationship with the Papahanaumokuakea Marine National Monument, and we've been doing this up there. So, our work has been able to span across all the islands. What we do personally tends to be a lot here and in the monument, but we do everything we can to make sure that all the groups on Maui and all the other islands have these resources too.
Thank you. There's several people asking that. We appreciate that. We had somebody wanting to say fantastic presentation and incredible work. What projects or site work are ahead for Mega Lab in the next year? What is your biggest challenge right now?
So I feel that like on the science front, the ball is rolling and we're doing really well there. There's obviously some uncertainty right now with federal funding and how that's going to go. So we're all paying very close attention to that. A lot of our work is federally funded through like National Science Foundation and in these partnerships with these federal agencies. But looking bigger picture, like we want to at least, well, I can definitely say we all want to, and very much me personally, at the end of my career, I want to look back and know that I hopefully left a positive mark and influenced other people to go into science. So my kind of larger goals that I'm trying to use Mega Lab to promote is just get kids excited about science and understand that like there's careers here and we need people doing these jobs, and it shouldn't always be people from outside or other places, right? Like we do a ton of work with kids in local schools, and every single time they stump me. They ask me questions about what they see and what they know about fish, and I'm always like, man, you guys are probably smarter than I am, because they're spending their lives in these environments. And I always couldn't stand how I was told often going through this career, look, there's no jobs in science, there's no money in science, so don't, think about something else. But here I am, right? And the people telling me this had jobs in science. And so it never made sense to me. And now that I'm in this space, I see jobs everywhere. We need people in conservation groups, in NGOs. The state is hiring people all the time. These agencies are hiring people. So I don't see it that way. And if there is a shortage, then we need to do everything we can to show how important this is and make more jobs. I don't think that tourism in Hawaii should be our biggest industry. I think our biggest industry should be protecting our valuable resources. So I really want to promote science in the economy, and that's where like the media and all these products, we hope it gets people fired up about science. And that's why we're also trying to be really ambitious with citizen science. You
Yeah.
don't have to be doing exactly what I'm doing. You could be making videos, you could be doing a whole array of work that supports this. And just like the live streaming camera, we've got thousands of people that just sit there, and there's a timestamp document. So anytime you see, like when that monk seal was sighted, they're writing it into a spreadsheet and they're producing data so that we can track everything that we're seeing and showing up. And that is super valuable. So I just want people to realize, like, it's never helpless. We need everything that we can get, and everyone can play a role, you know? And I think if we break down those barriers of what it takes to be a scientist, we realize everyone is a scientist. We're all curious. We all ask questions. So as much as I love the technical stuff and it's fun to get publications, you know, I don't want my work to be papers or, you know, digital papers on the internet. I want it to be people that hopefully maybe got a little bit inspired, and maybe that's why, you know, they end up going into this space too.
So good. So good. A few more questions if we can try and get through them. We've had the same thoughts. I think this was about AI. That AI is the future of large scale reef surveying. What do you see as the role for human divers moving forward? Because you did mention that there's a a place for all of it. So what do you, in your opinion, what is that place for human diving?
Yeah, I mean, so AI is so good at very many things, but it's important to not anthropomorphize it. So, you know, like for instance, Chat GPT, all these tools, you know, people are always talking like, oh, it's going to get consciousness, it's going to do this. As crazy complex as it is, it's a text compiler. So it takes text and it just does an amazing rapid job of extracting information. That text doesn't mean anything if a human being with creativity doesn't interpret it and use it for something good. So we can put all the wildest technical tools out there. It's not going to make decisions. It's not going to bring creativity to the table. And a lot of times your eye is the best tool you have on reefs, you know, and I really mean that. Like we've done crazy work looking at how AI can like auto detect diseases on corals and find things. It never is as good as a human being, and it doesn't have the same pattern recognition that we do. And I know I'm giving really long answers. I'm sorry about that, but you know, as an example, like when you drive to work, you know, you don't have an overwhelming panic attack being like, oh my God, a tree, oh my God, a car, a person, a this, a this, this. It's because the human brain is the best thing at rapidly detecting patterns. And you're just like plants, cars, trees, autopilot. And so that pattern recognition and creativity will always give people a role because we will always have an ability to see and understand things that computers just, I don't think, will be able to replicate.
Super encouraging, especially because AI is the big, I mean, in every industry all around the world, right? Everyone's like, yeah, that's going to take my job in 2 years. Not quite, not so fast, not so fast.
Yeah.
Another question, I hope I'm understanding this correctly. Is it possible to learn or estimate how many fish are needed for a healthy reef?
Yeah, we kind of basically take baselines from what we know, right? So this is why things like the Northwestern Hawaiian Islands as a monument are so important because there's no humans there, right? There's no fishing pressure. And so what that gives us is the most unbelievable lens into how should this food web look when it's able to operate on its own.
Mm.
And if you talk to me or anyone that's been there, they'll always start telling you shark and alua stories, right? Because you get there and you dive in and it's like, oh my God, I have seen like one alua in the last 2 years in the main Hawaiians, and now there's like 100 around me, and there's this shark and that shark. The reason is, like I was trying to mention in the talk, the energy's moving up the food web. That's where it's supposed to go. So those locations show us like this is what a healthy reef is supposed to look like. The energy is supposed to flow upward, and we have a big abundance of things at the bottom, but it rises to the top. And we have all these predators that are allowed to have their role. So those pristine sites are so critical for us. This is why I'm so passionate about, you know, preserving the monument, because without that, right, we don't have the baseline, and
Yeah.
then we can compare other locations to that. So, you know, we're as doomy and gloomy as it can seem at times, there's still tons of very pristine reefs around the world, and we need to protect those because that's what reminds us of how they're supposed to be.
That makes so much sense. Do you have any artists on your team who depict reef life for conservation reasons?
Yeah, we really try to partner with artists, athletes, you know, people that make media. And so we've got like even the little cartoon on the bottom here. This is an artist that we've worked with. And we are all about it because to us, artists are the same as scientists, right? It's just two sides of the creative spectrum, and there's often far more overlap than people give it credit. So I really love working with artists and creatives because they challenge me and they make me think differently. You know, and a lot of times the way they can interpret things artistically gives a whole new view and perspective on these systems. So a lot of times it's just us making fun, recreated things through art. But there's also a big role for artists in like drawing and depicting these complex data things, right? Like we're always so embedded in like boom, 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 the craziest technical graphs. And it's like, to a science nerd, that's cool. But a lot of times you need artists to distill that down into something that's meaningful that connects with people.
Yeah. Yeah, so true. Okay, last one, kind of a longer question, but so Maui's blessed to have a lot of different groups of people that are in the water at all times, recreational divers, tour boats, club ocean swimming groups, and they see these reefs every day and know them best. How would you think that we could go about utilizing them and their knowledge in capturing the data about the changing reefs? We want to have their information and contributions be purposeful and contribute toward larger goals. I think you had kind of mentioned this when you're talking about even free divers, um, being
Yeah.
a possible
So
source.
I totally agree. I think all those people have a really, solid knowledge about these systems are out there every day. And so that's why we're really trying to push these citizen science initiatives so that there's ways for them to contribute. So that whole study we did with the camera, we're really trying to like, because scientists can also be kind of, I don't know, exclusive a little bit. Like I have this degree, and I mean, look, I have the degree, and I'm proud of it, but it's still a piece of paper. I've learned more from honestly, I learned more from some like of the old fishermen that I fish a lot, and I've gone out with. I feel blessed to have gone out with a lot of the uncles here, and they have taught me so much more
Yeah.
than almost any scientist that I've ever met because they're so in tune with the environment. And so finding ways for them to be able to contribute to me is critical. So we did a lot of that just to validate like if you know your fish, you know this reef, and if you know this reef, your information is highly valuable. So part of it was that, right? Validating that voice, validating that skill. And what you mentioned in the very beginning, map to adapt, that's the mindset behind that whole initiative. So we're working on right now building a big website where anyone can contribute any form of data about a reef. And so it can be something as simple as a picture. We even want to make it as simple as just a text observation, right? Because it's logged now, it's documented, it's somewhere that can be referenced. And on the technical side of it, there's all these like free tools now. Like if I had seen this when I was in grad school, I would have probably, my head would have exploded. But there's tools on your phone now. So people can just put their phone when they're diving or have a GoPro, take a quick video of a coral, and it can automatically get three-dimensionally reconstructed online, and you can share that on the website. So we can get any of these data products from the most simple to the most complex. Anyone can add to it. And that's why we're ambitiously trying to get 1 million reefs. Because if 1 million observations come through, that just shows how powerful all these people are that are out on the ocean and how much they can contribute. And then on the back end, what we're trying to do is build a network so we can take these characteristics like color, reef complexity, very simply analyze them from something like a photograph or one of those 3D models and plug it into a network with all the other data that all the scientists in the world are collecting. So if you're on Maui and you're on the East Shore and you really like this one spot, take that picture and then you can see, hey, this is what other reefs it is similar to, and this is the ones it's different to. So then if you're wondering like, well, how is it doing? You're able to leverage all that science data too and say, okay, cool, we're really similar to these other reefs on the other islands. Maybe they're doing great, that's awesome, or maybe they're having issues with losing parrotfish. So let's keep an eye on that here. So we want to build that like two-way street where all those people can contribute and be having the conversation with us and making positive change together.
Yeah. That's awesome. My God. Wow. A lot to think about you've given us to think about today. Thank you so much, John, for sharing with us tonight, for graciously staying on and answering all of our questions. It sounds like you've inspired a lot of people tonight and given us a lot to think on in terms of, how are we what are the tools we're gonna use and employ to protect our reefs and our futures. So yeah, mahalo for being with us, John. We're so appreciative of your work. We also want to thank our partners one more time for helping us to bring these talks together. And a recording of tonight's talk will be posted to YouTube. Sorry, we couldn't. Facebook didn't like us today, so we couldn't live stream it, but we will post a recording so that you can go back, share it with your friends, revisit all of the great points that John made tonight. So thank you all so much for being with us here. Thank you, John. If you guys have other questions, he did list his email, so when the recording goes up, you'll have that information. And until the next time, everybody, thank you so much. Thank you, John. Once again, stay safe
Thank
in
you.
Hilo, and we hope to see everybody again soon. Aloha.